Hello students, today we will study chapter number 6, Seeds, Structure and Germination. Students, in previous chapter of pollination and fertilization, you have studied about double fertilization. You have studied that one of the male gamete fuses with the egg and form embryo, and the other male gamete fuses with two polar nuclei to form endosperm. You have also studied that after this process, the ovary converts into fruit. It means fruit is a ripened ovary. The ovary wall forms the fruit wall. Fruit wall is formed from ovary wall only, and the fruit protects the seed inside it. Inside the fruit there is a seed, so it gives protection to the seed. Now these seeds are ripened ovules. Seeds are ripened ovule, and these ripened ovules contain embryo inside them. It means they give protection to embryo. And this embryo, when gets the favorable condition, give rise to a new plant. There is one more word that you might have commonly heard, that is called grain. Grain is actually a fruit in which the fruit wall and the seed coat are fused with each other. So fruit wall and seed coat both are fused together to form a protective layer. Then we don't call it fruit, we call it grain. So in this chapter we have to study about the seeds, their different types, and about their germination. So let us first see classification and structure of seeds. Seeds, some of the seeds are very small in size. They can't be seen by naked eyes like the seeds of poppy or strawberry. And some seeds are quite large like mango seeds, plum seeds. You know that the largest seed is of coconut and double coconut. So their size vary. Now we come to the type of seeds. Seeds can be classified on two bases. Number one is on the basis of endosperms. If endosperm is present in a seed, then the seed is called albuminous seed or endospermic seed. Endospermic seed means endosperm is present in it. Students, you have to learn examples of these. Monocot albuminous seed, cereals and millets, they all are albuminous seeds. And in case of dicots, poppy and custard apple, they are dicot albuminous seeds. Second type of seeds are X-albuminous seeds, which are also known as non-endospermic seeds. From the name, it is very clear that they are devoid of endosperm. Endosperm is not present in them. Again, you have to learn some examples in case of monocots, orchids, in case of dicots, gram and pea. So this is the first basis on which the seeds can be classified, on the basis of endosperm. They can be classified into two categories, albuminous and X-albuminous. Second is they can also be classified on the basis of cotyledons present in them. If only one cotyledon is present in the seed, then the seeds are called monocotyledonous. Mono means one. Examples are maize and grasses. If two cotyledons are present in a seed, then the seeds are called dicotyledonous. Di means two. So examples are pea, gram or bean. Now we have to study about the bean seeds and about the maize seed. You might have seen bean seeds. Bean seeds, you might have seen this type of seeds. These seeds are, what is the shape of this seed? It is kidney shaped. Okay. All seed, there are different type of beans you used to eat, but the basic structure of all the bean seeds are same. Their shape is kidney shape. They have one convex side, this is convex side, and one concave side, right? These seeds are protected by a seed coat. Seed coat consists two coverings in it. The outermost covering is known as testa. This is a hard covering, very thick and hard covering, that is called testa. It protects the seed from any injury or any type of infection. Inside this testa, there is a thin layer that is called tegmen. It is the second layer of seed coat, that is called tegmen. It also has a protective function. It means it also gives protection to the seed. Now the next part of the seed, you can see here, a white color oval structure is visible here. White color oval structure on a concave side. This white color oval scar on the concave side is known as hilum. It represents the spot where the ovule was attached to the ovary. This ovule was attached to the ovary, right? Because this is ripened ovule. The seed is ripened ovule. So when it was attached to the ovary, it was attached to this point. So this scar is indicating that from here it was attached to the ovary wall. This scar is called hilum. Now just near to this scar, there is one pore present, a very small pore present near the scar. This pore is called micropyle. This micropyle is a small opening. It performs two important functions. Number one is that water enters the seed through this micropyle, and with this water the embryo swells up. Seed swells up. Second is that through this micropyle the exchange of gases takes place. So respiration in embryo is carried out through this small opening, that is micropyle, through which it exchanges the gases. Now inside this there are the cotyledons. Inside this there are two cotyledons present, so it is a dicot seed, and inside to that there is a embryo. Embryo has a structure like this, right? So two cotyledons are attached, and this portion is a embryo portion. This is embryonic axis. Now this point where the cotyledons are attached to this embryonic axis, the portion above this means this portion, जहाँ cotyledon जुड़े हैं उससे ऊपर का portion, this portion is known as epicotyle, right? This portion is called epicotyle, and this part is known as plumule. This plumule give rise to the shoot system, and the part below the point of attachment of cotyledons, ये नीचे वाला हिस्सा, it is this axis, portion of this axis is called hypocotyle, and this portion is radical, which give rise to the root system.